Hi, good afternoon, good day everyone. Today we're going to have our Science 8 Quarter 4, specifically on week 2. And today we're going to discuss one of the basic concepts in science, or specifically in biology, that would be mitosis and meiosis. Um, my name is Juline Joy A. Taro, and I am your Science 8 Grade A teacher in the Asa National High School. So let's start. What are the most essential learning competency for this video is that number one, we're going to compare mitosis, meiosis, and their role in the cell division cycle. Next would be explain the significance of meiosis in maintaining the chromosome number. For this video, we're going to have a learning objective that would be define chromosome, cell division, mitosis, and meiosis. And our second learning objective that would be compare mitosis, meiosis, and the role in the cell division cycle. And the third one is explain the significance of meiosis in maintaining the chromosome number. Now, for the cell division, we're going to first identify what is cell division all about. So I have here two pictures that best depicts what is cell division all about. That would be cell division is the process by which cell divides into two new cells. Take down note in your science grade 7, you have already discussed one of the basic concept of biology that would be the parts of the cell. Take the note that cell is the basic unit of life, and every one of us, every living things in this world is made up of cells. As humans, we have billions and trillions of cells inside our body. So, why do cells need to divide? Why do it need to divide? Number one reason that would be living things grow by producing more cells, not because of each cell increase in size. So they need to divide in order for us to grow. If you can still remember when you are still a baby, you're still a toddler, you're already 100 centimeter, 60 centimeter, and you turn out to be 160 centimeter, that process of having increase of height is considered as the process in cell division. Next would be repair and of damaged cell. Damaged tissue. Take the note that if you still have cuts on your body, on your hands, and then you notice that your hand is healing, automatically the process of healing those wounds is called as the cell division. Next is, if cell gets too big, it cannot get enough nutrients in the cell and waste out of the cell. So our body, we consume food, and those food are converted into different kinds of nutrients. So those nutrients are automatically microscopic. They are very, very small, and those nutrients, those microscopic nutrients, will be absorbed by microscopic also observing mechanism. So that cell is consuming microscopic nutrients, and they also excrete waste product microscopically. So they should be a little smaller. It will not turn into a bigger cell, so that it will circulate, it will absorb and give out microscopic substances needed for within our cell activities, or we needed in our cell body. Next is, original cell is called as parent cell, while the two new cells are called as daughter cells. So I have here a picture which best depicts what is all about. I have here a parent cell, and I have here a two daughter cells. Take the note that before cell division occur, the cell replicates all of its DNA, so each daughter cell gets complete set of genetic information from the parent cell. Before it ends up like this one, the cell undergoes a process by which it ensures everything that is inside the parent cell will receive by the daughter cell. So it undergoes different processes in the pictures. And for example, this one is DNA replication. At this stage, they undergo mitosis. This process ensures that everything inside this parent cell will also be present in their two daughter cells. And these two daughter cells are identical to one another. It means that whatever genetic information which can be found in this daughter cell will also be present in this daughter cell. So this daughter cell is considered or called as two diploid cell. Take the note, two diploid cells in cell division is considered as identical. So each daughter cell is exactly like the parent cell, same as the kind of chromosome as the original cells. So whatever number of chromosome find again in the parent cell will also be the same in their daughter cell. So in a unicellular organisms, reproduce by means of cell division, which is called asexual reproduction, and that is called as binary fusion. So like this one, this is actually a paramecium. No. This is actually a bacteria there's the binary fusion, and it turns out that the division will result into reproduction. But this reproduction is called asexual because there will be no exchange of gametes. So it is called as asexual reproduction, specifically only on unicellular organism. But fortunately, we are not unicellular. We are multicellular organism, which means multi. We are made up of many of cells, many cells. That is called multicellular. Next, we're going to define what is DNA. So DNA is located in the nucleus and controls the cell activities, including cell division. If you can still remember, in the parts of the cell, that nucleus controls all the cell activities. That is why the reason why the nucleus is the one who controls it. It is because it contains the DNA. The DNA is the master of all commands inside the cell. So, long and thread-like DNA in a non-dividing cell is called as chromatin. So, and double-coiled short DNA in a dividing cell is called as chromosome. So, I have here a picture. So, actually, this is a DNA. Part of this one are DNA. And then, once it coils up into a protein like this one, the circle protein, that one, you will name it histones. Actually, this is a histone protein which is coiled up together with the DNA. And once they coiled up, it is called as double-coiled short DNA dividing cell is called as chromosomes. And this chromosome, when they are coiled to one another, they are coiled into different to one another. This is what it looks like. This is a chromosome. Okay, so consists of chromatid and a centromere. This part is the chromatid, and this one is the centromere, which is the attachment of the chromatids. Okay, now chromosome structure. So for this one, for singular chromatid is called as chromatid. When they are duplicated in the process of before the cell division itself, they have been duplicated and they are called as identical chromatids. Like this one, duplicated. Okay, when the cell divides, sister chromatid separates and one goes to each cell. So the reason why these are duplicated, so that when it undergoes cell division, this will split up, and this part of this chromosome will will be received by one daughter cell, and this part of this chromatid will be received by another daughter cells. So chromatin like this one. So why does DNA need to change from chromatin to chromosome? So why is it that from this one, they need to coil themselves, they need to uh, coil themselves and then duplicate into this one? Why is it important for them to coil up? So it would be more efficient in division. So take the note, it's like you're going to a vacation. So before you go to a vacation, you need to what? You need to pack up your things. You need to put it in one place. You need to put those things in the bag. Same also with the chromosome when it undergoes cell division. They need to fix the things. They need to pack the things so that when they go outside, they go from one place to another. They will not forget anything. They have their jam-packed objectives, or they have their jam-packed things that they need to their cell activities. Okay, understood? So, the chromosome number, every organism has its own specific number of chromosome. So, each organism, like this one, the baby, the dogs, the fish, and the cabbage, these are considered as living organisms. Each of them have this specific numbers of chromosome. Example, for human, we have 46 chromosome. So paired up together, they are, or we have 20, pair up together, this 46 chromosome pair up together, we have 23 pairs of chromosome. The dog, like this one, the dogs have 78 chromosomes. Paired up together, we will have 39 pairs divided by 2. So, I have here the goldfish. The goldfish have 94 chromosomes. Paired up together, we have 47. And the lettuce, or the cabbage, I said it's lettuce. It has 18 chromosomes. Paired up together into 9 chromosomes. Now, all somatic cells or body cells, they are called somatic cells, in organisms have the same kind of number of chromosomes. So, example, human have 46 chromosome. Human skin cell have 46 chromosome. So, human heart cell 46 chromosome. Human muscle cell have 46 chromosome. So, that is the reason why. So, the chromosome inside my head is 46. The chromosome inside my eye is 46. The chromosome inside my ears is 46. Paired up, they are 23. So, each of those numbers are equal to the numbers of chromosomes inside our body. Okay? So, the fruit fly, they have 8 chromosomes. So, the fruit fly skin have 8 chromosomes. It's again, their heart have 8 chromosomes, and their fly muscle cell have 8 chromosomes. So whatever the number of chromosomes they have equal amount of chromosomes in each part of their body, in each of our body. Okay? So I have here a cell cycle. What is cell cycle? A series of events cells go through as they grow and divide. So I have here a picture which best describes the cell division. So the cell grows, prepare for division, then divides to 2 daughter cells, each of which then begins the cell cycle again. So I have here the cycle, or this is the life of the cell actually. So at this part of the cell, we call it as the first growth phase. So you will start at this point, so the growth and normal metabolic roles. The next stage will be this stage is called as the G1. The next stage will be the S phase. So at this stage, the DNA is replicated. Take the note, this DNA contains all the genetic material inside our body. Next, the G2 phase, we have the growth and preparation for mitosis. So the cell is actually preparing itself to undergo the division, the mitotic phase. So it undergoes different kinds of um, preparation so that the cell will be ready to undergo this part. Okay? So, take down note that the G1, the S phase, and the G2 is considered as the whole phase of interphase. That is called interphase. So, what is interphase? It is the period of self-growth and development. So the DNA replication is at this part. During the interphase, the cell also grows, carries out the normal cell activities, replicate all organelles. So its cells spend most of its life is called as the interphase. If you can see on the picture, the biggest lifespan of the cell is in their interphase stage. When they are equivalent to this lifespan. They have a bigger lifespan in the interphase stage. So now we will go to the mitosis. What is mitosis? It is the division of nucleus into two nuclei, each of the same numbers of chromosome. Now, mitosis occur in all somatic or body cells. Take the note that somatic cell is considered as all the body cell. The body cells, the cell in my forehead, the cell in my eyes, the cell in my hands, the cell on my neck, the cell on my feet, they are called as somatic cells. Again, what is the opposite of somatic cells? We have here are the cell in the reproductive part, that is another kind of cell. So any part of our body is called a somatic cell, and this mitosis occur at this stage. So why does mitosis occur? So the reason for the mitosis to occur is so each new daughter cell has nucleus with a complete set of chromosome, like the picture I have given to you. So I have here four phase of nuclear division, mitosis directed by cells DNA. And the shortcut for this one is actually PMAT, so that you can remember it um, automatically. So the first phase of nuclear division is called as prophase. Next is the metaphase. Third is the anaphase or the apart, and telophase is 2. So let's discuss it one by one. So what can you see on the picture? What is happening at this stage? Again, this is prophase stage. So the chromosome coils up. As you can see in the picture, diba, the histone is coiled up into them. Next is the nuclear envelope. The nuclear envelope is this one. It disappears. The spindle. So I have here prophase. What is happening in prophase? Number one, the chromosome coils up. So if you can still remember so I have here a picture of the prophase. So let's identify what is happening in prophase. So number one, chromosome coils up. So as you can see here, the chromosome starts to be bold, and they are the chromosomes are coiled up with the protein histones. Next is the nuclear envelope disappears. So actually this is the nuclear envelope. Before the cell division, they are actually connected to one another. But when the cell division process tries to start up, this automatically melts down, tears down into pieces, and it automatically disappear later on. Next is the spindle fiber forms. So these are the spindle fiber. Later on, you will know what is the main role of this spindle fiber. The next stage of the cell division is the metaphase. Metaphase is actually the meaning of meta is called as is identified as middle. So what are the things in the middle? The chromosome line up in the middle of the cell. So actually this chromosome starting now to go to the center of the cell. And the spindle fiber connects to chromosome. So these are the spindle fiber. They are trying to connect to each of those chromosome. And the next stage will be the anaphase. So the anaphase, which means apart. So what is happening in anaphase? The chromosome copies and divide. So they divide, and they are actually pulled by the spindle fiber. Spindle fiber pulls chromosome to opposite poles. So the chromosome which is attached in here, they are pulled to one of those poles. And this stage is called as anaphase. Next is the telophase, which means two. So the chromosome uncoil, the nuclear envelopes form again, silently this chromosome, and then the two new nuclei are formed, like this one, two nuclei, and the spindle fiber disappear. So there's no spindle fiber anymore. Next is the cytokinesis. Cytokinesis is the division of the rest of the cell or the cytoplasm and organelle after the nucleus this divides. So in the cell cytoplasm pinch in. So the division of the cytoplasm will start from the outside. Unlike the plant cell, the cell plates form from the inside, going to there. So after mitosis and cytokinesis, the cell returns to interphase to continue to grow, perform regular cell activities. So this is an example. There will be cleavage furrow. Oh, this is a cleavage furrow of an animal cell, and this is the cleavage furrow or the cell plate formation in plant cell. So you can see in animal cell, the cleavage formation or the formation of this cleavage, the force is from the outside. While in here, in the plant cell, the force of division will start from the inside. So that is the difference between the animal cell and the plant cell. Next will be we will have a summary of the cell cycle. So we have here from the interphase. So the interphase, we have here G1 S phase and the G2 phase. And next will be the mitosis part, or the things under this mitosis is the PMAT. And the next stage is the cytokinesis, which is the division of the cytoplasm itself. Then it will go back to interphase, and the cycle will not end there. It will continue to uh, undergo from different process to one another. So when cell becomes old or damaged, they die and replaced with new cells. So like in this picture, so when they are damaged, they reproduce, they divide so that they will grow another one. So the cell division controls I have here. A DNA controls all cell activities, including the cell division. Some cells lose their ability to control, so their rate of cell division, the DNA of this cell has become damaged or changed or mutated. So the superdividing cells masses are called tumors. So benign tumors are not cancerous. These cells do not spread to their part of their body. So malignant tumors are cancerous. So So I have here a short review of the things which is happening in I have here a small table that you can fill out, and you can pause the video if you wanted to. You fill out the information in a separate sheet. on what is happening in the chromosome appearance and location and interphase, and what are the important events in the interphase. Same as with the prophase, you write the appearance and the location and the important events. You can pause this video and do it for 5 minutes. Once you are done, so let's discuss. Okay, so in interface, let's have a short review. So the interface have the DNA copies itself, and the location of the chromosome is inside the chromatin. What are the important events? So we have here the different, you can actually pause the video if you want, and fill out the important details in the table. So I have here different phases in the cell division, the process of the cycle of each stages of the cycle. So I have here the different stages of the cell cycle. And we have here the interface and the process we're in the cell division, of course. So I have here the column for the chromosome appearance and the location of the chromosome. And what are the important events which we can find in this each life stages of the cell. So you can pause actually the video and try to fill out this table, and um, you can have it for 5 minutes. Once you are done, uh, we will fill out the table for a short recap. So in an interface, we have here the DNA copies itself, and the location of this DNA is inside the chromatin. What are the important events? The DNA replication, the cell grows and replicates of organelles. So take the note in the interface. There are 3 stages there. We have the G1, the S phase, and the G2. So this events is under these key stages. Now let's go to prophase. What is the appearance of the chromosome there? The chromosome actually coils up, and important events: nuclear envelope disappears, spindle fibers form. So those are the important events in prophase. In the metaphase, the chromosome line up in the middle. Take the note, the other name for metaphase is the meta, which actually means in the middle. And the important events in the metaphase will be the spindle fiber connects to the chromosome. Next, in the anaphase, we have here the chromosome copies, divides, and aparts. So, moves apart actually. The spindle fiber pulls the chromosome copies apart to opposite poles. So, take down note that actually uh, nga naman! So, I have here the different stages of the cell cycle. We have here the interphase and the process for in cell division, of course. So, I have here the column for the chromosome appearance and the location of the chromosome. And what are the important events which we can find in this each life stages of the cell. So, you can pause actually the video and try to fill out this table, and um, you can have it for 5 minutes. Once you're done, so I have here in the anaphase, the chromosome copies, divides, and moves apart. So the spindle fiber pulls the chromosome copies apart to opposite poles. Now next, let's have here the telophase. The telophase chromosomes uncoil back into a chromatin. And the important events are the 2 nuclear envelopes reform, new nuclei are formed, spindle fiber disappears. For the cytokinesis now, I have here the chromosome is in the chromatin, and the division of the rest of the cell, division of the rest of the cell, the cytoplasm, and the organelles. So I have here in the anaphase, the chromosome copies, divides, and moves apart.